It's often said that adaptive evolution, the natural creative process so powerful that it gave birds their wings, mud skippers their hop, and snapping turtles their nearly perfect fish-luring tongues, it's often said that adaptive evolution is one part random mutation and another part non-random natural selection. Random mutations provide a continuous supply of variation, some good, some bad. Natural selection acts as a filter on that variation, allowing beneficial mutations to accumulate while continuously erasing all that is bad. Over many generations, this surprisingly simple two-part trial and error process, it can build spectacular adaptations. But what exactly do scientists mean when they say that mutations are, quote, random? Furthermore, what's with the stream of headlines, sometimes even popping up in the journal Nature, claiming, at least in the title, that mutations are not random? Has there been a fundamental shift in how we understand evolution? Or is this a bit of a word game? Stated Clearly presents, what is mutation bias? What does it make you think of when you hear the phrase, mutations are random? Simple logic tells us that mutations can't be completely random because, well, there are only 4 standard types of DNA nucleotides. Sure, the nucleotide cytosine can be mutated, can be transformed or swapped out with thymine or guanine, but it can't be swapped out with, say, an elephant or a tesseract, right? Well, if that's the case, what other unspoken limitations might exist for these so-called random mutations? As a quick refresher, a mutation is any change in the sequence, the order or number, of nucleotides in an organism's genome, in an organism's chains of DNA. Mutations can be small. A single pair of nucleotides is added, deleted, or swapped out for a different pair of nucleotides. Or mutations can be large. Entire stretches of DNA can be duplicated, moved, horizontally transferred from a different cell, or even deleted. Mutations are caused by many things, including copying errors during DNA replication, normal wear and tear from everyday use, damage by radiation, chemicals, insertions from viruses, the list goes on. When a scientist says most mutations are random, what they often mean is that the organism did not detect or somehow decide which new mutation it wanted next, and then actively cause the appropriate mutation. No. There are a few exceptions to this, but most mutations happen to an organism, whether that organism likes it or not. Random does not mean, however, that all mutation types are equally likely, or that all regions of the genome mutate at exactly the same rate. Mutation biases do exist. Every now and then you'll find a sensational headline suggesting that mutation bias is a completely new revolutionary concept, but scientists have known about mutation bias since at least the early 1960s. That said, it is true that recent advancements in genetics are allowing us to study mutation bias with far greater clarity, and some researchers have claimed to find that natural selection can exploit mutation biases in really interesting, unexpected ways. But that's a topic for another day. For now, why is it, in general, that mutation biases exist? Well, DNA is a physical thing inside a physical cell. Parts of a DNA chain are simply more fragile than others, and some regions of DNA are better protected by the cell than others. As a helpful, but of course an imperfect analogy, this here is a string of punched tape from an old Soviet computer. It contains an early version of computer code. Each line of holes and non-holes acts as a byte of digital information that can be read, one byte after the other by a machine, similar to how in a protein coding stretch of DNA, each set of 3 nucleotides acts as a codon that can be read by a ribosome. Here I'm going to pull on this tape until it breaks, but before I do, can you guess where it will break? Which byte or bytes of encoded information do you think make the weakest links? I'm gonna guess this one right here, but let's find out. I was slightly off, but I was pretty close. The point I'm making here is this. When information is encoded into a physical object, either by a computer programmer or the natural process of evolution, it can have the side effect of dictating the durability of that object. This is the case with old Soviet paper tape data files, and of course, this is the case with DNA. Mutation rates can differ wildly throughout a cell's genome at multiple scales. If we zoom in all the way to individual nucleotides, we find that cytosine, under many conditions, is more fragile than the other three types of nucleotides. Cytosine tends to mutate more often. Furthermore, transition mutations, the swapping of similar-sized nucleotides, these usually happen more often than transversion mutations, the swapping of different-sized nucleotides. Zooming out, scientists have found that E. coli bacteria have a wide mutational hot region, a region where mutations happen more often, that gradually gives way to a mutational cold region. It seems this is caused by the specific way E. coli handles its DNA when reproducing. Zooming back in, but not all the way, we find that inside those larger regions, small-scale hot spots and cold spots can also be found that depend on local DNA sequences. Here I built a physical model of DNA to show you how my favorite type of sequence-dependent mutational hotspot works. But first, let's do a quick refresher on base pairing. A chain of DNA is actually made of two strands stuck together by weak bonds in the middle, hydrogen bonds between nucleotides. Guanine is attracted sort of like a magnet to cytosine, and only to cytosine. Adenine is attracted to thymine. My wooden model here uses actual magnets to simulate this base pairing process. It only has two types of nucleotides instead of DNA's four, but this is enough to show how local hotspots work. Here, my red nucleotides are magnetically attracted to blue, but magnetically repelled by other reds. Inside your cells, DNA is constantly being tugged around by enzymes. It's often untwisted, and base pairs can be temporarily pulled apart in small stretches. Look what happened when this sequence was opened. Two nucleotides from the same string found each other and automatically base paired. Because of this, the DNA is now stuck open. This could cause problems, but in this case it's not a big deal because most of the other nucleotides did not find partners. It only takes a little tug and the entire chain will automatically snap back together. Well, almost. This model doesn't work exactly like real DNA, but you get the point. What happens, though, if we look at a different sequence of DNA? Here in the middle is what we call an inverted repeat, or palindromic DNA. If you take the sequence of reds and blues on the left side, make a copy and invert it, it is identical to the sequence on the right side. Watch what happens when I separate this sequence of DNA. Almost every single base automatically found and snapped to a partner on its own string. The cross that formed here, this is what we call a DNA cruciform, and cruciforms are extremely vulnerable to mutation. At the top and bottom of the cruciform, we have nucleotides that could not reach their partners. They are exposed to the elements, they can be easily damaged or mutated. Furthermore, there are several types of protein complexes that wrap around DNA and zip along its length. This way of traveling is important to their function. When these complexes hit a cruciform, they can crash, derail, or stall, sometimes also causing mutations. Inverted repeats are just one of many examples where a specific nucleotide's chances of mutating depend on its local neighborhood. The nucleotides just upstream and just downstream. Natural selection, of course, can then promote or demote any of these mutations. Okay, so what does all of this mean? Are mutations random? Yes, but the word random here must be carefully understood. Most mutations are random in the sense that they do not happen, quote, on purpose. Cells usually don't decide which specific mutations they want and then actively cause them. That said, mutations are not perfectly random. Some mutation types are more common than others, some spots in the genome are more fragile than others, some regions of the genome are more carefully protected by the cell than others. If you work in statistics, you might say that mutations do happen randomly or stochastically, if you want to sound extra smart, but mutations have a, quote, uneven distribution. This, of course, is also true of most games of chance that everyone considers to be random. When rolling a pair of dice, for example, you are far more likely to roll a 7 than you are to roll a 2 because there are 6 completely different ways to roll a 7. Again, random, but with uneven distribution. Scientists currently debate how much of an impact mutation bias really has on evolution, but a growing number of researchers do agree on this. A stronger understanding of mutation bias will help us make better sense of evolutionary history and make better evolutionary forecasts in the future. That said, despite what we read in sensational headlines, mutation biases do not change the fundamental rules of evolution. Natural selection remains the primary sculptor of adaptation. I am John Perry, and that is Mutation Bias, stated clearly.